So as you continue through your experiments for your biologically important molecules, the last three are going to be for each of the others, right? So in the last video, we talked about carbohydrates. So here we're going to do one experiment for each one of the last three. So 4.3, you're going to start with lipids. Now, lipids are a little different than the rest of our biologically important molecules in that they don't have a common monomer. They are all quite diverse in their structures and their makeups, but the one thing that they all have in common is that they are hydrophobic, which means that they have bonds that are sharing things equally, right? They're non-polar covalent bonds. So lipids have no common monomer, but none of them react with water, right? That's why they get put into this category. So in order for us to test a lipid, we have to use another nonpolar thing, right? There's this whole thing in chemistry, they talk a lot about like dissolves like dissolves like. So a nonpolar molecule can dissolve into another nonpolar molecule. So the test we use is using a powder called Sudan 4. So the Sudan 4 powder is nonpolar and it can mix with other nonpolar things and other hydrophobic things. So it works really well for us to get a response here so we can recognize if it's nonpolar. So with this test, you're going to use the same test tubes that we use for all of the tests, but you are going to use a powder instead of a liquid here. So the Sudan 4 is a powder. And because your fingers are also hydrophobic, it will stick to your fingers, not for long, but it will stick to your fingers. And if you're anything like me, you'll end up with it all over your lab book during this experiment as well. So to do this, you're going to use a toothpick in order to pick up a little bit of powder and just dump it into the test tube. Then you'll swirl the test tube and you'll look to see if if the powder actually dissolves into the liquid or not. You will compare it to your controls. So again, you'll have a lipid control that will have lipid in it, and you will have the negative control, which is going to be the water. So then you will compare those test tubes and you will decide, okay, does this have lipid in it or not? So let's look at those two controls here, right? So again, the first tube is our positive control. The Sudan 4 has dissolved into the fluid and has made everything red all the way throughout, right? So this is a positive reaction to the Sudan 4, meaning there is a hydrophobic lipid molecule here. Now this is an example of what will happen when it does not dissolve. Notice that there's clumps all over the place, right? This is the Sudan 4 clumping to itself because it cannot interact with water. So this is what you will see if it is not hydrophobic, if it is not a lipid. So you may see clumps kind of form along the top of the fluid, or some of them may fall down to the bottom, but you will not see that dissolved nature all the way through. So one thing I will note here, so for most of our test tubes, like I said earlier, we will be reusing them, you'll be washing them, and it will be great. We'll be able to reuse them and not contribute to landfills. But with these, we can't, because the way the Sudan 4 and the lipids interact with the test tubes, they're basically trash after this. So you'll see a very specific collecting material for everything for Sudan 4, and then you will put the test tubes into a waste container that we will take care of separately. It's not possible for us to reuse these, so keep that in mind. So next step is our procedure 4.4. So procedure 4.4 is going to be for proteins. So proteins are made up of a monomer called amino acids, right? Amino acids are a monomer with a very similar shape, right? So this top image here is the amino acid. There is a carbon-hydrogen base. Then there's an amino group, right? So this is one of the functional groups we talk about in lecture. And then there's a carboxyl group, which is here. That is true of all amino acids. But what changes is this side chain. So the side chain is going to be different in each amino acid. So when we put these amino acids together, we again go through a dehydration synthesis reaction in order to form a peptide bond. Now, a peptide bond is a covalent bond. It's just a fancy name for a covalent bond found in our proteins. And that is actually what the test we're going to be looking for tests for. It actually looks for this peptide bond, and that is what we'll test for. So when we build proteins, and again, in lecture, you've talked a little bit about primary structure, secondary, tertiary, and quaternary. In order to form the primary structure of a protein, we have to put these amino acids in order. And that's something you'll talk a little bit more about later on in the semester when you are building proteins using transcription and translation, which is going, yeah, that'll happen probably a little bit later. But to understand that the amino acids get put together in a different order, that's one of the things that gives proteins its specific function, right? Because structure determines function. So if you have a specific structure and order of these amino acids, those amino acids will fold up in different ways, and that will give it a different structure, right? So the amino acid order is going to determine that structure. So if we mess with that structure, which is something that can happen, and again, you'll play with this a little bit more in Lab 5 when we talk about enzymes, because enzymes are a type of protein. Proteins can be basically taken apart by denaturing these bonds. So protein structures can be denatured and then become non-functional. Now, what is kind of interesting is the most basic form of denaturation. It does not involve the peptide bonds. It actually involves the hydrogen bonds that are holding everything together. So the hydrogen bonds will be interfered with. We'll be testing that in Lab 5. So proteins have a specific structure that starts with a peptide bond and then progresses through other types of interactions in the molecule, including hydrogen bonds and hydrophilic and hydrophobic interactions. And then we put more than one of these chains together to form a really nice 3D shape. If we break that 3D shape, we break the function of the protein. So if we denature the protein, we break the function of that protein, which means it won't be able to do its job. So in this procedure, you are going to be testing for the peptide bonds using a burette or biuret reagent. So for this one, you're going to be adding 2 mL of the solution that's in table 4.4 with 0.5 milliliter of that reagent. It just has to sit for a little bit, view it, swirl it, look at it, and again, compare it to our controls. So compare it to the protein control and our water as the negative control to determine which one has the peptide bonds and which ones don't. Where are our protein molecules? So let's take a peek again at those controls. So here we have our positive control on the left. Right, so it turns a purplish color. Now again, remember, the darker the purple, the more peptide bonds that are in here. So this is a relatively light purple. There are not that many peptide bonds in this solution. Then over here, nothing happened. All right, it's kind of a blue color, right? You have a light bluish color, which is going to mean there's no peptide bonds, there's no proteins in this solution. So by comparing to these two, you can determine not just if peptide bonds are present, but even how many peptide bonds are present, right? Is this a lot of protein or a little bit of protein? So the last of the procedures you're going to do in this lab is looking for a specific nucleic acid. So I said earlier, nucleic acids are not something most people have heard a ton about as a category, but you've all heard of DNA, and DNA is a nucleic acid. So in this test, we're actually going to be looking for DNA. So all nucleic acids are going to be made up of monomers called nucleotides. So nucleotides are always going to have a similar structure as well. So they'll have a specific type of sugar, right? So in DNA, this is deoxyribose. It will have a phosphate group that is attached to it, and then it will have what's called a nitrogenous base. So if you remember anything about DNA, you know that it has bases that are A, T, C, and G. Right? Now those bases all have names. For example, A is adenine, it's not just A. So these are going to change based upon what type of nucleotide you are looking at. But the structure is still the same, which is why we call it a monomer. So in order to build DNA, we have to put these monomers together. These monomers will get put together with a dehydration synthesis the same way that we've talked about with all of the rest, right? They all get put together the same. We will see the bonds occurring between the phosphates and sugars. So it'll basically be a sugar, then a phosphate, and then a sugar and a phosphate, and that will continue all the way down the length of each side of the DNA. So we showed this picture earlier, right, where I was talking about hydrogen bonds. So all of the like backbone they call here, right, of phosphate, then a sugar, phosphate, sugar, those are all covalently bound together, dehydration, synthesis reactions. But the bond in the middle between the bases, so this is base 1, base 2, base 3, and base 4, they will hydrogen bond together in order to give us the structure of DNA. Okay, so there are different types of bonds that have different types of functions here, right? So the covalent bonds are a little stronger for more permanent structure. The hydrogen bonds are actually here to be broken so that we can pull DNA apart and put it back together again, right? Which is something you'll talk about again as you look at DNA replication a little bit later this semester. So in this lab, you are going to be looking for DNA using a dish diphenylalanine test, or what I usually just call the dish test. I've heard other instructors call it the DD test. So whatever you call it, same test. This is going to use 2 milliliters of the solution. Now, in order to get this reaction to happen, we need a little bit more than just a swirl it in view, right? For this one, we do have to boil it. So in the lab, we actually do this underneath the hood because it smells very vinegar. It's a little volatile as it boils. So it's all done under the hood in hopes that that smell and those molecules will get taken out of the classroom by using that covered hood. So we're going to boil these test tubes for 5 minutes, and then you're going to take those out and throw them into ice. So again, remember, hot things are hot, so be really careful here. This happens to be the experiment where I got a really awesome burn that has left a scar on my forearm because I forgot hot things are hot. So, you know, be careful. So you boil the water, and then you boil the solution, and then you throw in ice for 5 minutes. And then again, you're going to compare. So you're going to have a DNA control, and then you're going to have a water as the negative control, and you will determine if DNA is present or not. So in general, if there's no change, right, which this is the negative control, it will be negative. So it'll be clear. It might be a whitish, foggy color depending on what you're testing. So for example, if you're testing the albumin, which is egg, it will get kind of cloudy because it was cloudy to begin with. Then if there is DNA present, it's going to be blue. So again, just like we've talked about in previous tests, the darker the blue, the more DNA that's there. So if there's a lighter blue color, it's still positive. It just means there isn't as much DNA present in that solution. Okay, so if you have the PowerPoints of this, here's kind of the general overview of everything. The test the biological molecule is using, what we're looking for, and then what color it will change. Now, the other thing you will also be doing in this lab is looking for unknowns, which means you are going to have an unknown solution, like I said earlier, unknown A or unknown 2, however we have it labeled, where you're going to test that unknown with every single test. So you're going to put that through all 5 tests, and you're going to look to see if it turns positive or not, right? So does it have starch in it? Does it have DNA in it? You're going to be running those tests to determine and kind of apply what you're learning here. What molecules will be present if we test for your Bio-Ret test. So as you look ahead towards your homework assignments and your quizzes and your tests in this course, for this specific lab, you are going to have to know what test is testing for which biological molecule. You are going to need to know what those colors represent, right? So if you have a Benedict's reaction that turns blue, does that have reducing sugars or not, right? So you should be able to apply all of this information given specific unknowns on a quiz or an exam.